Okay, once you are on this screen, let's come up here and let's change the name. Let's make this yours. So click, drag, and highlight copy of, and we're going to change that to your name and 8C. So go ahead and put your name and your class up here on the top so that I know this belongs to you, not to somebody else. And then once you've got that typed in, we're going to move it into your science folder on Google Drive so you can find this later. So right here where it says move, how do you change it? So all you do is you click on the title up there, and then you can highlight. So click and drag and highlight anything you want to get rid of. So it originally said copy of. So I'm going to highlight that and just start typing over it. And you're going to put your name and then 8C. If you want to be cool like me, you can put your first initial and then your last name. No, I think your last names are unique enough in this class that you could do that and I'd still be able to figure it out. So that's up to you. Definitely put 8C. And then just click anywhere else and that will save the name. So to put it in your folder, does anybody remember how to move this into a folder and would possibly like to tell us how to do it? If so, please raise your hand. Christian, do you want to tell us how to do it? I will try to unmute you. You can accept it or deny it.
Sure.
All right, tell me how to do it.
So you would go into that little folder icon you said.
Okay.
And then you would press the 8C science.
See if I have
with
one.
I think it has our name on it.
Okay, yeah, I think most years I was able to change your name. So I'm gonna put it 8B just because.
And then it's gonna ask a question about whoever has access to this will be able to see it, and just put yes.
Okay, awesome. So mine doesn't say that because I'm not sharing it, but yours all will say that because I had you guys share it with me at the beginning of the school year. Awesome. All right, so does everybody have that moved into a folder? If so, just visual on your cameras, give me a thumbs up. All right, got a couple thumbs. Excellent. All right, so let's go ahead and move to slide number 2 then. So modeling mitosis in a fruit fly. For those of you who were doing the lab on Friday, there was a little blurb of information at the beginning, just introducing it. And it tells us, you may have seen fruit flies buzzing around a bowl of fruit. They are tiny, but if you look closely, you may see their red or white eyes. Like all living organisms, fruit flies grow and reproduce. The diploid number of chromosomes in fruit flies is 8. The diploid number of chromosomes in fruit flies is 8. In this investigation, you will simulate mitosis and meiosis in fruit flies. Through modeling, you will learn how fruit fly sex cells end up with only 4 chromosomes. All right, so I went ahead and turned this into slides. And on slide number 2 right here, it's telling us modeling mitosis in a fruit fly. The diploid number of chromosomes in fruit flies is 8. Go ahead and type into the chat, anybody who knows. What does a diploid number mean? Okay, I've got one answer, and I need to learn how to not typo and instead try typing for once. That might be something new, Wriggle. Okay, so I'm hearing total, I'm hearing group of chromosomes, number of chromosomes. What else could it mean? And I'm liking all these answers. I'm not going to say which one is correct. They might all be part of the same correct answer. Okay. Okay. Let's see. Okay, So pretty much everybody who's giving me an answer is definitely mentioning chromosomes. I'm hearing all of the chromosomes, double the chromosomes, total, and group of chromosomes. So chromosomes, chromosomes, chromosomes. So when we're talking about just any old regular cell, oh, has 2 copies of each. Ooh, nice. Imani just gave me a good one. Every cell in that organism has 2 copies of each type of chromosome. Just got one, a full set of chromosomes. So in any cell in your body or in an organism, you have 2 copies of every chromosome. So like in this one right here, I just moved in a black and a yellow. So it got one of its chromosomes from mom and the other chromosome from dad. And that would be from any sexually reproducing organism. And so the diploid number means that it has 1 chromosome from each parent. The opposite of that would be the haploid number, which means it only has half the chromosomes because it only has the chromosomes from mom, or half the chromosomes only from dad. Yeah, so the diploid is the full set of chromosomes. I'm going to steal Giselle's words on that one. So your diploid set does not necessarily mean the double amount of chromosomes, but it does mean that it is twice as many chromosomes as you might expect in a gamete. It would be all the chromosomes together in one cell. So what we're going to do on this page right here is we're just going to move each of these sticks right here. Each of these sticks is a chromatid, and it represents a single chromosome. We're just going to move them into the cell. And go ahead and place these on your slides where you think they would go in the cell. Are they going to go out here in the cytoplasm? Are they going to go in the nucleus? Are they going to be like half and half? Go ahead and move them where you think they belong. And if you want to get fancy and you want to like rotate them, I think you have to have 2 or more. You can do that, and you can spin them, put them in, and you can go kind of diagonal if you want. You know, just do what you want. And just move them all in, and they all belong in this purple area, which represents the nucleus. Okay, so you should all have 8 sticks of chromosomes, 8 chromatids up there in your nucleus. Okay, I'm going to go ahead and move on to the next slide. So these are the first 4 questions from the lab. What is the diploid or total number of chromosomes in a fruit fly body cell? Go ahead and everybody type into the chat what number it should be. And I can go back to slide number 2 if it helps. Diana got it? Yasmin's got it? Oops, I don't want to move that. I want to do that. Let's see. I've got 2 people telling me the number of homologous pairs, but there we go. Perfect, Martha. Thanks for that correction. 2 people have given me the homologous pairs number, but we're looking for the diploid number. So the total number of chromosomes. All right, perfect, Christian. Thank you. All right, so our answer to number 1 is 8. There are 8 chromosomes in the fruit fly, in each cell of a fruit fly. So how many homologous pairs are there? Diana's got it. So the homologous pairs would be every group of 2 of the same type. So this black and this yellow right here that are the same size, these are the same type of chromosome. So they could be considered together as a pair. They're a homologous pair. These 2 right here are the same size chromosome. They're the same type of chromosome. Fatima's got it. Same type of chromosome. They're a homologous pair. These 2 right here, they're the same type of chromosome, same size and everything. One came from mom, one came from dad, but they are a homologous pair, because they're the same type. And these two right here, again, homologous pair because they're the same type. So I would have 1, 2, 3, 4 homologous pairs. Another way you can think about that is if you have 8 socks, how many pairs of socks could you make? You can make 4 pairs of socks. All right, so use the diagram. You can zoom in on your screen if you would like. So grab the little zoom in magnifying glass over here and just click with your left mouse to go in, right mouse to go out if you went too far. And figure out which ones are the names of the parts of mitosis. Okay, now don't just list every single phase off. That's cheating. There should only be 4 steps. So everything on this diagram is part of the cell cycle, but we are looking for just the steps of mitosis. So you can ignore the part where the cell is just like doing its thing, being a cell, doing what cells do. You can ignore the part where it actually splits off into 2 completely different cells. Mitosis is only the 4 steps where stuff is actually happening. So which are the 4 steps where mitosis actually happens? Go ahead and type them in, or you can type them onto your slides. Perfect, Diana, thank you. Oh wait, the step before your last one. So the C1 is not part of mitosis. All right, so I'm seeing cytokinesis showing up. That is actually after mitosis. So mitosis is the time where the cell is doubling itself and moving all those chromosomes around. But as soon as you have a full intact cell with only the diploid number of chromosomes, then you're back into the normal cell cycle. You're out of mitosis at that point. Okay. Perfect, Austin's got it. And then again, Diana and Fatima are super close. Giselle, you're missing 1 piece. Kylie is very, very close. All right, so the only parts that are actually part of mitosis, it starts with prophase. So it starts with prophase. That's where the DNA doubles, the chromosomes form, and they are twice as many, and that nucleus starts to go away. Next is going to be metaphase. So I'm going to delete. So metaphase is where all of those chromosomes start to line up in the center. Anaphase is where they start to kind of pick a side. They start to kind of migrate away from each other, and they'll be far away. And then at the last part is telophase, where all those chromosomes are on complete opposite ends of your gigantic deformed cell, and they're getting ready to convert into the 2, but they're still 1 cell at this point. So interphase is the time in between mitosis and not mitosis, basically. Interphase is the intermission, the time where the cell is just relaxing and doing its thing. So prophase is where everything doubles. Metaphase is where it lines up. Anaphase is where it starts to migrate away. Telophase is where the nuclei reform. And then after all 4 of those steps, that's when it breaks up into 2 completely different cells in cytokinesis, and then it restarts the cell cycle at interphase. Okay, so which 4 steps are part of the rest? The hint is, it's the 3 we did not use. So we didn't use interphase. That's where the cell is just existing. Interphase, cytokinesis equals when okay, so let's see. Cytokinesis is definitely part of it. Interphase, late interphase, and cytokinesis. Perfect, Christian. And also Yasmin got it in as well. And let's see. Telophase is still part of mitosis because you still have that one gigantic cell that's starting to get ready to break apart. So interphase, late interphase, and cytokinesis. So if you have these in your slides right now, then when you go back to your lab on Go Formative, you can go ahead and correct your answers. I tried to keep all these numbers the same as they are on Go Formative. Okay. You guys ready to move on to the next slide? Okay. I'm gonna go ahead and go. Okay, so the next slide is, nope, okay. Back and tracking. Okay, sorry. I went back. I'll give you guys about 20, 30 more seconds. Unless there are questions. If there's questions, please ask. Otherwise, just a couple more seconds. Okay. All right. 5, 4, 3, 2, 1. Blast off. All right. So we are going to demonstrate interphase again. So if you remember from the very second slide, we moved all of our chromosomes into this cell. This cell is an interphase. It is just doing what it does. So we're going to do the same thing right here. So just go ahead and grab these little sticks and arrange them inside the nucleus however you see fit. And I'm just going to keep them paired up with their partners. Okay, so interphase, all we have is the diploid set. Not the duplo blocks that you might play with if you want to have like the gigantic Legos. We're talking about diploid, not duplo. Okay. So on this page right here, I would like everybody to type to me the answer to number 5. How many cells are there in interphase? Hint, count how many cells are on this slide. So how many cells are on this slide? Our cell is being represented by the big white circle with the little kind of light purple on here. So we have 8 chromosomes, and those 8 chromosomes are inside of how many cells? Perfect. Yasmin, Giselle, you both got it? Alyssa got it. Noemi got it. Ashley, a little smaller on your number. So I have 8 chromosomes inside of how many cells are on this page? This big white circle is a cell. How many big white circles are on the page? I'm going to have to go back and remember how to count. How many, so right here where my mouse is moving around, how many big white circles are there? There we go. Now I'm seeing some ones and oh, one. There is one cell right now. That one cell has 8 chromosomes in it. So how many cells are there in interphase? We only care about one cell when we're talking about mitosis. It could be happening anywhere else in the fly. It could be happening anywhere else in any other cell in the organism we're talking about. But when we are explaining the mitosis process and we're explaining the cell cycle, we only care about one cell at a time. It is way too complex to try to consider every single cell. Look at your own hand. How many cells do you think are in your hand? Now think about your arm, your whole entire body. How many cells are there? I am not about to think about every single cell in my body as I'm talking about mitosis. I'm just going to focus on one at a time. So the answer to number 5 is one cell. So in this one cell, how many chromosomes are in the nucleus? Now we can say the other answer that everybody wanted. Let's see. Christian's got it. Yasmin's got it. Giselle's got it. Fatima's got it. So we are not asking for homologous pairs. We're asking for chromosomes. So just count the sticks. Let's see. Diana's got it. Kylie, Lizeth, and Martha got it. Austin's got it. Awesome. Alyssa's got it. All right, so there are 8 chromosomes in this one nucleus, and Christian just got number 9. So 1, 2, 3, 4, 5, 6, 7, 8 chromosomes right here. So there are 8 chromosomes in each nucleus of our one cell. Okay, last question is, how many homologous pairs are there? There we go. So Christian already had it. Alyssa, Yasmin, Austin got it. Giselle's got it. Oops, that was meant to everybody. Copy. Noemi's got it. Martha's got it. Copy. Everybody. Kylie's got it. Okay, so our homologous pairs, remember, those are 2 of the same size, 2 of the same type of chromosome. So I've got 1 pair right here, 2 pairs, 3 pairs, and 4 pairs. Brian's got it, perfect. All right, can I move on to the next slide? Give me some thumbs ups. Just some visuals on your screen. Any questions on this slide? Okay, seeing some thumbs. I'm moving on. We're gonna keep on rolling. All right, so the next stage is prophase. Prophase is where all that DNA doubles up and it starts to form into X's. So it forms into chromosome pairs or homologous pairs. Actually, chromosome pairs. So we are going to take these sticks right here and we're going to arrange them in X shapes into our nucleus. So this is where they start to double up, and we're just going to grab one of each size of each color and make an X. Make sure when you're making the X's, they're of the same color. So what's happening is they're just doubling up the DNA they got from each chromosome, and that chromosome copies itself and like mirrors itself, but it's kind of bent into an X shape. You're not going to have them mixed with a black and a yellow. So just go ahead and grab each of these and start arranging them however you want. You can get a little creative if you want. If you highlight 2 pieces and grab this little circle up here, you can twist it around. But in the idea of saving time, you can just pull them in. Make sure they all fit in that purple area. Make sure they all fit in the nucleus. And then so on the lab for Friday, what you would have been doing right here is you would be using your poster board where you drew out your diagrams, and you would take the pipe cleaners or the yarn or the twist ties, whatever you ended up finding, or paper even for that matter. So like I had my paper chromosomes that I held together with tape. You would be taking these and putting them into your diagram on the prophase section. So like I would go here on prophase, and I'd put all my X's into my prophase. But we're just going to do that right now in the slides. And I'll give you guys a couple more seconds to finish that up. So we went from our individual 8 sticks to now we have 8 X's, or 8 homologous pairs. But there are 16 chromosomes now. There are 16 chromatids. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Okay, I'm going to move on to the next slide. So the next slide is metaphase. I'm just going to grab all these X's by making a big box around them. I'm going to pull them out just so it's easier to find them. So metaphase, who can tell me how we need to arrange these X's? And you can raise your hand and I'll call on you, or you can just type it into the chat. How are the X's going to be lined up for metaphase? Can they be random, or is there some special specific way to do it? Perfect. So I'm seeing across the middle and the center. So just grab each of these X's. I locked them together this time. So you just have to grab one and then just start arranging them around the center. I've got an answer about the middle. Perfect. It doesn't really matter if they're all in color order or size order, just as long as they all fit into your cell and they're going across the middle. So I'm gonna like switch it up a little on mine, try to get some visual creativity going on. There we go. So just line them up in the middle. You can do it straight up and down, or you could have them go across this way. Maybe make them take some more space in between. And again, it doesn't matter how they're ordered. It just matters that they are all across the middle. So you can do that. You can do it that way. You can go diagonal. I don't care. Creativity is okay in this one, just as long as they go somehow through the center. Basically make a diameter with all of these chromosomes. Okay, I'm going to move on to the next phase unless anybody is not ready. Okay, so after metaphase is anaphase. So I've got all my chromosomes lined up in the middle from metaphase. And now what we're going to do is we're going to start pulling those chromosomes apart. So if I go back to my Cheerio picture, right there. Right. So at this point in anaphase, we're going to start pulling them apart. So the centromere is going to go away, and these chromosomes, one is going to go to each side. So my X comes apart, and they go to either side of the cell. So in this time, I do not have the X's linked, so you can just grab one piece and move it over. And if you want to make sure you're only grabbing one of each to go to each side, you can do like I'm going to do, and I'm just going to make sure that anything that's slanted, let's see, slanted downward to the right, I'm going to move it to the right side. If it's slanted downward to the left, I'm going to move it to the left. That way I can just make sure I'm pulling one of each, one of each, one of each, just ripping them apart. Destruction. And when you're done with this, you should end up with your diploid number again. You should end up with 8 chromosomes on each side. If you do not end up with 8 chromosomes on each side, you may need to double check. So I just took those X's and I ripped those chromosomes apart. So I have single chromatid chromosomes on each side now. And they can still be kind of random-ish. They just have to make sure that they are on opposite sides. And then I'll just take some visual thumbs up when most of you guys have this. And just double check. I have 1, 2, 3, 4, 5, 6, 7, 8 on this side. 1, 2, 3, 4, 5, 6, 7, 8 on that side. 4 yellow, 4 black. And I can move them around and make sure they're the same sizes. Yep, those are the same. Those ones match. These ones match. And those ones are supposed to match, but they don't really. Hey, look, now they match. So just make sure you have your 8 on each side, and then we will be moving on. That looks cool. All right, I got a couple thumbs ups. So we're gonna go on to the next piece. Okay, so the next part is telophase. So in anaphase, we just ripped those chromosomes apart and pulled them to their separate corners. It's like, gotta get you away from each other. You guys are going crazy. We do not want this fight to continue. So we're in telophase, and that's where the nuclei starts to reform. So all these chromosomes need to go into the nucleus for each side of this kind of double cell right now. So I'm just going to take my sticks from here. I'm going to take a big one of each color and move it into my nuclei. Let's go there and over there. Then I'm going to take the next size and the next size and its pair and its pair. So just go ahead and pull all of your chromosomes in so that you have perfect diploid sets again. And if you're waiting for me, you can like grab these and rotate them. You can leave them straight up and down, however you think looks good in yours. Okay, so telophase again, we had them already over on their separate sides because anaphase just ripped them apart. And then telophase, we're rearranging them into the nucleus because we are about to split up. This is the very last piece of mitosis, right before the cell goes back to being normal. So we do not have a cell membrane right now, or a nuclear membrane, I should say. So it's not quite 2 cells joined together, but it's about to be. Can I move on to the next stage? So mitosis is now done. We now need to have 2 complete perfect cells. And that's how cytokinesis is going to do. So I'm going to move my chromosomes into my cells again. And if you were following along with the materials instead for the lab, You could just like literally slide them to the next stage. But this way we get to play with fake digital sticks here. There we go. All right, so however you make yours look is fine, just as long as you fit 8 in the left cell and 8 in the right cell. And then we're going to ask the questions in just a moment. Okay, so remember, when we started the mitosis, we were only caring about one cell. We only had one circle representing the cell on the screen at any given time. One cell, one cell. How many complete cells do we have now at the end of cytokinesis? Perfect, Alyssa got it. So how many cells are there now that we're finished? Again, you're using the number of chromosomes. Yeah. Christian's got it. Think a little smaller for the 2 who just sent me an answer. How many white circles are on this page right here? How many cells do we now have that are complete? There we go. Now I'm getting some correct answers. Diana, Yasmin, Fatima, Samantha, Laura, Isabel have both told me correct answers. And then Diana just changed it for some reason. Bruh, no. Let's see. Yeah, so let me respond. Yeah, so we started out with one cell during interphase. It doubled all of its information. It lined up its information. It pulled its information apart, and then it aligned that information where it belongs, and then it created 2 perfectly identical daughter cells. So at the end of cytokinesis, there are 2 cells. Okay, so question number 8. Sorry. Number 8, how many? Okay, so question number 8, how many chromosomes are in each nucleus? Okay, Yasmin and Alyssa have got it so far. Oops, and then I just like typed it in. So if you were paying attention, you've already got the answer. So count how many sticks are in each of these nuclei if you made it correctly. 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. Perfect. Diana just gave me a correct answer. In only one cell, though. In only one cell, everybody. Perfect. There we go. So Diana has it. Fatima has it. Isabel has it. Martha has it. So there are 8 chromosomes in one nucleus. So 8 chromosomes per nucleus. It doesn't matter that we have 2 cells now, because it wants to know in each nucleus. So this one cell right here is just like the original cell that we started with, because it has 8 chromosomes in it. And if it's just like our original had 4 homologous pairs, and Christian just told me the correct answer. How many homologous pairs are in each of these cells? Yasmin and Alyssa got it. And let's see. Kylie's got it. Austin's got it. Emmanuel's got it. Martha's got it. And I've got an answer that's too high and an answer that is too low. So our homologous pairs are, oops, why is that not clicking? There we go. So our homologous pairs are pairs of our chromosomes that are the same size, the same type. So this black and this yellow right here are one pair. This black and yellow are your second pair, third pair, fourth pair. So we have 8 chromosomes, which makes 4 pairs. Diana's got it, perfect. So we have 4 homologous pairs. So again, we have our 1, 2, 3, 4, 5, 6, 7, 8 chromosomes in this one cell right here. Those 8 chromosomes make up 4 pairs. 8 divided by 2 is 4. So we have 1 pair, 2 pairs, 3 pairs, 4 pairs. Okay, over here I've got 1 pair, 2 pairs. I gotta make kind of like a little happy face. Can I grab just both those? So these 2 right here, oops. These 2 right here make up 1 pair, 2 pairs, 3 pairs, and 4 pairs. Are there any questions on this page? Does that kind of make sense? I still have 1 or 2 incorrect answers that have not been fixed yet, but most of you have given me the correct answer in here. There we go. Giselle just gave me a correct answer. All right, if there are no questions, I'm going to go ahead and move forward.